Hi, my name is Suzanne ORorke and I'm a second year PhD student here in the NCBES in NUI Galway. In May 2007 I graduated from NUIG with a degree in biomedical engineering, and the following October I returned here to pursue a PhD in the biomaterials group under the supervision of Professor Abe Pandit. The overall aim of my work is to use a gene therapy approach to treat a disease known as recessive dystrophic epidermolysis bullosa, or RDEB for short. RDEB is a skin disease where blistering occurs on the skin and on internal linings such as the intestines. It can occur in varying degrees of severity, so one patient may have very slight blistering in a restricted area, whilst another may have extensive blistering over their entire body. It is caused by mutations in the type 7 collagen gene. Type 7 collagen is responsible for the formation of the anchoring fibrils in the skin. The anchoring fibrils help to keep the layers of the skin intact, so when these fibrils aren't formed correctly, as in RDEB, the skin layers are more fragile and blisters form. RDEB is an autosomal recessive inherited disorder. This means that both parents carry the abnormal gene, but neither is affected with the condition. RDEB patients have a high risk of developing other diseases such as skin cancer. By the age of 35, over 50% of RDEB patients will have developed skin cancer, and most will die within the following 5 years. In my work, we're focusing on a method to deliver the normal gene to the keratinocytes. Keratinocytes are a type of cell found predominantly in the skin. An average keratinocyte in the human body is about 10 to 30 micrometers in size. If we want to deliver something into this cell, it must be much smaller than this. As a result, we focus on nanosystems. 1 nanometer is a millionth of a millimeter. This cannot be seen by the naked eye. Instead, we use very high-powered microscopes in the lab. The nanosystem we propose to use are called nanoshells. The nanoshell is formed using different biodegradable materials. The gene is encapsulated in the hollow core of the shell. The outer surface of the shell is then functionalized using a dendrimer. Dendrimers are hyperbranch polymers where each branch can be used to add small molecules to the shell, so to improve its function. In this case, I am adding a fluorescent probe so that we can track the nanosystem. I will also add a homing factor so that we can direct the system to the keratinocytes. It is hoped that the completed system will then be delivered to the RDEB keratinocytes. The gene will be released and type 7 collagen will be produced normally again, correcting the disorder. An important part of the work is the formation of nanoshells. Within the biomaterials group, many people are working together to produce these shells using different materials, including chitosan, collagen, and polyethylene glycol. In this method, nano-sized polystyrene beads are used as a template to build the shell around. The beads are coated in the selected material. After, the polystyrene can be dissolved, leaving the hollow core shell. These shells are then viewed using a scanning electron microscope. Unlike regular light microscopes, SEM uses an electron beam which allows us to visualize samples at very high magnification. The SEM in the NCBES has a magnification range from 30x to 500,000x. In this image, the nanoshells can be seen clearly under high magnification. Another aspect of my project is the functionalization of the nanoshell surface. When these systems are placed inside the body, they must be tracked. To do this, I want to attach a fluorescent probe to the nanoshell through the dendrimeric system. The fluorescent probe used in the study is called fluorescein and is more commonly known as FITC. It is chemically conjugated to the dendrimer molecule. It is then incubated with cells so we can establish how it may act inside the body and whether or not it will be effective. In the lab, we can visualize this system in the cells using a confocal microscope. The confocal illuminates fluorescent samples, therefore the probe can be viewed easily. In order to view the cells also, we stain them with different dyes. In this image, the actin cytoskeleton of the cells has been stained red with rhodamine phalloidin. The green fluorescence is the dendrimer probe complex. This work is supported by Deborah Ireland and the Department of Biomedical and Mechanical Engineering here in the National University of Ireland, Galway.